He is Fellow of the National Academy of Science and Fellow of Indian Chemical Society. He has 12 patents granted and more than 170 publications, 10 monographs, and 5 books chapters. He guided more than 15 PhD students and 35 postgraduate students. His field of expertise includes drug development, pharmaceutical formulation development, and regulatory toxicology, targeting various disease and health issues. He received numerous awards, including the Gandhi Young Technology Innovation Awards from the Biotechnology Industry Research Assistance Council, Laboratory Scientist Awards, Technology Awards from the Defense Research Laboratory, DRDO. Today, we are really privileged to have him as our guest. Now, I would like to request Dr. Biplab Kumar De, Dean Academics, to felicitate our honorable guest. Please, sir. Again, I would like to request Dr. Biplab Kumar De, sir, Dean Academy, to come upon the stage to facilitate Himeshwar Goswami, Professor Emeritus. Please, sir. Thank you so much, sir. Now, I would like to request Dr. Panavesh Chakravarty, sir, to take over the dais. Please, sir.
Slide. So start. So very good afternoon. The front of us, the excellent of that audience. Especially, I ought to give many thanks to the organizers, especially Dr. Biplab sir, Dr. Goswami sir. Yes, obviously, your Dean of Pharmacy is also a good friend. All my dear some students that are with the complete PhD under me, they are in faculty, and they are all lovable faculty and dear students. See exactly what you mean that this is discussions in point that is the nutraceutical could be treated as a drug or as other things that can be food or other thing would can be you promote your health. So some embarcations is there, some narrow boundary is there, and this boundary is give the meaning actually. Being in the one of the defense researcher, our food definitions are what in different in cases. We think that survivability is our food actually. In when go to any defense people will ask in the when does surviving in the jungle, surviving in the war, that is how we can survive in the food. That is their food actually, what they have been getting. Today, many things is coming here, and technology, even though I have the many limitations actually of the topics that I could not discuss. Do you think when you go to the Chandrayaan missions, it is coming in the our next primes, our Chandrayaan missions, India is going to the launch actually. So what will be the food in Chandrayaan missions? If you think that tomorrow we will go to the in the Chandra, in moon actually, what will be the our food? You cannot not still you cannot define. Yes, this food can be in the tablet forms. It is many questions will become again. This yes, if you take the tablet instead of food, that will be sufficient food to you. Let's will come the next slides. And food means in Bhagavad Gita there is one fantastic two lines actually. Just sister Sita Santo Mukhinanam Sarva Kilancha Munjana Satta Papaya Pastana Karan Karana. The spirituality minded to eat the food that is the first offered by the sacrifice and released from the all kind of sin. Others who can food their own enjoyment, variety it's only the same. That is basically food actually. In Bhagavad Gita, basically this is defined actually food actually. Even when the food is a Brahma in the Hindu mythology, and whatever the Brahma means, it is the creature of this all the universe. So we have taken the Brahmarpanam, Brahma भी ब्रह्मानुतम, मतलब हम ब्रह्मा को हम अंदर ले रहे हैं, ब्रह्मा को पचा रहे हैं हम खाने, मतलब ब्रह्मा को ही पचा रहे हैं. That is mean food, and this is give you energy. So let's become some interesting, some demarcations, food and nutraceutical, your health supplement and drugs. So next slide, please. Some point actually. Now sometimes food used as a medicines. Now today concept is coming that now it is for the baby food, it is for the geriatric patients food, it is a kidney failure food, it is for the sugar and diabetic food. Many food is coming. But that is known as a father of medicines and what is called the Hippocrates. So several years ago, the let's food to be your medicine. The philosophy behind the focus on the preventions of the particular disease. Next. So basically, if you think, I can even go to the accommodate to Hindi also, right? Hindi and English both you can speak. I think there is nothing wrong. So, बचपन में मैंने कभी कभी देखता हूँ आप लोगों को और मैं आपने तो सोचता हूँ बचपन में व्हील चेयर की बात करें जो हम when we were a very small child actually, then what happened, our mother or parents taking the to in particular trolley and they are taken in the art actually. When become your oldest people, you will do the same, you will get the trolley actually, you cannot walk actually. So in starting and ending will be the same. So बच्चे आयामी धरती में the peoples have not come and लास्ट मौत होने के पहले तक हम दवाई के ऊपर ही भरोसा करते हैं. So nothing but the food become the drug actually. So basically you are taking this one. Next one, please. Nutraceutical can define as a food or part of food or nutrient that provide the health benefit, including the prevention of the treatment of the disease. Particular in disease treatment, that's a nutrition is in use. Next. Now, many functional food is there. In the definition, if you go to the particular act or law, in 2021, that's a draft guideline has came. It is implemented in 2020. It is additional to the FICC Act in 2006 actually. They have taken the many definitions. What are the health promoting? That in dietary supplement, it is a functional food, but there are some many regulations will become and many boundaries also there. So I will today discuss some point actually. Fatty acid, then we source in milk and milk and meat, improve the body compositions and reduce cancer. That is the fatty acid DHA. And EPA, basically the fish oil, bear seed, maize, powder, mustard seed, rapeseed oil, basically you give this type your PUFA or free fatty acids basically. Reduce the cardiovascular disease and improve the mental and the visual health. Next. Like polyphenols category, anthocyanidin, it is from the fruits. Catechins from the tea and bubble pot, mustard cake, rapeseed, and sulfate. It is good to neutralize the free radicals and reduce the risk of cancer. Flavone from the citrus, flavones from the also the flavonoid from the citrus, and flavones from the fruits and vegetables and soybean, and phytocynin from the cocoa and chocolate tea and rapeseed, and reduce the basically cardiovascular disease. So basically, the polyphenols is one of the largest target of the compounds of any of particular interest of. I've seen the many literature that in the polyphenol. Even tea has a very good polyphenol. So polyphenol has some roles, including the your edible oils also, including the mustard and essential oils also. Next. Now, fourth categories comes probiotics, prebiotics, and cyanobacteria. Lactobacillus, it is come from dahi and yogurt. That is principle of the basically gastrointestinal health. Fructooligosaccharides, whole grain, onions, and combinations, and and prebiotics. Phytosterol, that is dietitian, diastinic, basically soybean, flax, lentils, maize, budscreen, the leucine, the soybean soda, basically keep the phytoestrogens, reduce the menopause symptoms and increase the bone health. Lignans, flax, dyes, vegetables, reduce the cancer and heart diseases. Next, corticoids are carotene, like vesting, lignine, oat, maize, and fodder, carrots and vegetable fruits, neutralize the free radicals. Lutein, that vegetables and healthy sun, the fantastic the health actually lutein. Xanthocene come from the eggs and citrus. It is also increase the health results. Lycopene from the tomato reduce the prostate cancer. Dietary fiber like insoluble fiber for the heartburn, reduce the breast and the colon cancer. Alpha glucan from the oats. Whole grain from the cereal grains or reduce the severity. Next. And 7 number, market nutraceuticals, many market nutraceuticals coming in the different brand name basically. Like, betadine, carotenoid for the immune infections. Brand name is the Xanthor for the lutein ester, that's the eye health. Lipoic, that is alpha lipoic acids, the potassium antioxidant. Ginoniol from the phytosterol, that is the CHG reductions. Premium products like probiotic and intestinal disorder. Swalite, soybean cholesterol in the bone. Next. Now, it is interesting that drugs is basically vegetables or nutraceutical. If you take the carrot actually, and you see one carrot in actually basically see in the mirror. But carrot could not be your supplement of your all type diseases. This means that the vegetable pills we are not put in my nose. It was ready before I know in carrot because you cannot take carrot as a whole as a medicines in any cases. Next one. Now, the main attraction of this particular drug cosmetics act is if you know that 3D sections and the drug cosmetics act 1940 define herbal drugs. And section 22 of the drug and cosmetics act of 1940 defines the Ayurvedic, Siddha, and Ayurvedic drugs. So the molecules from the Ayurveda, Siddha, and Unani, when you were claiming as your nutraceuticals or as drugs, it could not be can adore actually in this case. Drug shadow, last slide. Drug shadow covers a white area. In this case, it's a white shadow. And nutraceutical sometimes comes in the form of tablet, capsules, liquids that contain vitamin and minerals and make certain claims. Just one example I can quote you. Do you think the pudinhara, it will be drugs, or then it is a drugs, or it is a herbal product? Can you define it? I think you'll be questions yourself. That pudinhara will become as a drugs, or it is in herbal products, or is a nutraceutical. With pudina oil you are using, but yes, it is in drug actually, because pudinhara tablets you'll get in the Dabur product, but it's in drug and cosmetic food, because you make the unit dosage forms. When any unit dosage form is comes, it is category as a drugs, and it is a necessary. It does not mean necessary you're taking from the plant as a food product actually. So you can understand the drugs and basic difference between the drugs and basically nutraceutical or the food. Next. Now, chapter 2, where is a food and health supplement is telling on. Main ingredient they are telling in the particular FSSAI Act. The schedule 1 for the vitamins and minerals, we cannot add more than that. Schedule 2, any forms of vitamin and minerals. Schedule 3 is amino acids. Vitamin 5 is plants and botanicals. Schedule 6 is a mineral or the metal. Schedule 9 for the enzyme as specified. So, when you do any nutraceutical research, does not you explore yourself, because it is already defined. Today what happened? We are doing many research, but not result actually not comes, and the people are not happy. They are not giving the fund to you also. Today government is very critical. So you can take your advantage that what the law says, and according law you can make your projects, and you can ask to the government of the funding, because they like to say their end product actually today. The today scenario is the end product. Today, your research is does not mean the publications only are patent. We are seeing like to get you the something comes. So generally, in this case, you can exit the schedules actually. What schedules is telling, take this ingredient and make it for the formulations. Caution should be given food and health supplement and not for the medicinal use. This product not intended for the diagnosis, treat, cure, and prevention of any diseases. Next. Chapter 4, it is known the nutraceutical category. In these cases, the phase nutraceutical would be written in the bold and readable in the label. So whatever you are taking as a nutraceutical, so nutraceutical claims should be there in the label, not more than improving the physiological. And this nutraceutical is increasing the physiological functions, but they cannot treat, they can't claim that is a cure of your diseases. So claims relating the disease improvement, but not disease cure. It can be the improve actually, but it cannot be cure actually. So this is another claim of the nutraceutical, because if you claim something, the 1951 Act is there for advertisement or provision of the Advertisement Act actually. So you cannot tell that it is cured by your nutraceutical. So trial with the human aspect, your clinical trial rule 2019 in this mandatory. When claiming that any molecule so like name of the product should be helping keeping your heart healthy and your heart healthy. So this claim will come from all of the clinical trial. So nutraceuticals should process the clinical trial basically, keeping your name and the particular product should helping you keep the liver function. So that is also in trial. This type claim you have to go to the clinical trial. Next one. Chapter 5, that is a food for special dietary use, excluding the infants, those to be taken under the medical advices. This food and special dietary would not be used in these cases. The formula of these types, total energy value should less than 800 kilocalorie or 3350 kilojoule, and not more than 1200 calories. Why? We will take the why this much. सिंपल क्वेश्चंस आपको हम पूछे, आप पूरा दिन में खाना में कितनी एनर्जी चाहिए, कितना एनर्जी तैयार करते हैं? अराउंड 1200 का एनर्जी चाहिए, 1400 से 1200 के पीछे 1400 to 1200. So this is mean this could not be exceed the value of your 1200. So limitations is they are fixed actually, that is 800. So the food or special dietary uses, there is some limitations, and the limits should be the levels should not be include any assertions suggesting that prevention, cure, treatment, same any particular disease or diagnosis or the disruption of the body natural physiological functions, whether temporarily or permanent. Next. Chapter 6, that is the food and special medicinal purpose. That is called the sometimes SMP Act actually. Nutritionally incomplete food, where the normal compositions or nutrient adapted formulation tailored or addressed to the particular disease, like diabetic food, right? And anti-diabetic food, like you cannot take the sugars much more. You can take the high dietary fibers. This is known as all are in the special medicine purposes food. It is called sometimes SMPP food actually. recommended to be used under the medical advice only. This is the label claim and nutritionally complete actually. This is basically nutritionally claimed foods actually. Next one. Chapter 7 actually, it is called the food containing probiotic ingredient. Remember, in the authority is approved only 2 probiotics, 2 category of the probiotics, not more than. So you cannot add any other microorganisms in this year's food. So only that lactobacillus cereus, I think, and another category is also there. 2 only the your microbes is allowed. Only probiotic cultures have been approved in the food authority. I think 2 they are approved. On that belong to the specific strain listed in Schedule X in the Drug Cosmetic Act, basically the FSSAI Act actually. Regulation are allowed. The categorical word, the probiotic food. Dahi yogurt is one of the common example. Only the prebiotic ingredient listed in Schedule and actually 11, and this rules as a prebiotic ingredient approved by the food authority. Next one. Now, some argument. Law and argument between this one. AdtU Pharmaceutical versus State of Kerala in 2nd April 2002 actually. This is a particular case where the vitamin E and vitamin C capsules. The multivitamin capsule is a dietary supplement they have claimed. The High Court of Kerala said that, Honorable High Court of Kerala said that substances are in the definitions of drugs under the Section 3B. And distribution, selling, and stocking should be follow the Section 8 of the Drug and Cosmetic Act. So you can understand the vitamins, vitamin E and vitamin C. Even if you're claiming in the unit forms, it will come the drug. So they you get the law actually. We are not changing even the vitamin C, maybe the nutraceuticals, but it is come in the category of drugs. Next one. Dr. Reddy's Laboratory versus the State of Kerala and 12 June 2008 basically. Another very good example. Dr. Reddy's Laboratory Limited was selling the gamma linolenic acid capsules, which was either treated the diabetic neuropathy, paying the tax at 12.5%. If you go to the nutraceuticals, the tax value is low actually. In this case, 12.5 actually, but GST basically. But however, assessing the authorities, the supplement would become the actually like Bonvita and Horlicks, which tax up to 20%. So if you get in the nutraceuticals, so it is increasing basically tax like the Bonvita or nutraceutical. So changing the name and they are getting a tax benefit. Ultimately, they have to pay the 20% tax. Next one. State of BR versus Alchem Laboratory. LPA, that is a particular 2009 case number. And Honorable Patna High Court directed the director DGHS, the Drug and Cosmetic 1942 report that the component mentioned in the label in the category food or drugs. Sometimes glucose and glucon D. The same phenomenon. Both are the glucose. That is in beta glucose. So if you take the glucon D, you take the microdname. It is somewhat different tax. Whereas in glucose actually selling the different, the tax reasons will be different. So that is one that all uncommon actually rules actually in cosmetic tax. Next. Proprietary food, that is the clearly example of the proprietary food, and execution such forms this act under the section. Section 1 of the 8. Packaging and labeling regulation, pre-packet food, and the consideration all packages food are governed by this clause. 6 and 22, all foods which are value-added properties such as nutraceutical, nutritional food, novel food, health supplement are categorized here. Next one. Chapter 9, and now, I have seen the many research actually. People are telling that is a plant drug and using as a food, and food is using as a plant. So this is basically clause in chapter 9. But the specialty food containing the ingredient question, Ayurveda, unique traditional medicine and system and Siddha. Remember, if you use the chemical ingredient from Schedule 5, 6, and 7 of the Ayurveda, Siddha, and Unani Act actually in diets, it could not be treated as a food actually. It became a drug actually. So you should avoid the section 5, 6, and 7 in these cases. The enlisted the molecules which are in the Drug Cosmetics Act by AyU's Act actually. AyU's rule actually. That is an act actually. So this ingredient could not form the plant or animal mineral or metal. It could not be processed or unprocessed. So no other ingredient that schedules 5, 6, and 7 could not be used. Next. Chapter 10 is a novel food. Preconditioning implied without approval of the food authority cannot manufacture to sell any trade implements. Both the manufacturer and importer have to require to submit the required documents along with the applications of the approvals. So now chapter 10 only the novel food category where we are also exploiting. mainly, I'm telling some classicals of the defense food, which are using for the defense only, like water ball. It's a simple water ball are used in case of water, mineral water, because actually the actual positions in the war or any scenario, we cannot carry the water bottle because a large number of volume. So they are using the water ball instead of this one, because ammunition is required much more, because the bag cannot accommodate the water bottle sometimes. That's time we are using the water balls, like high energy food, sometimes called have the maximum calorie one food. Air Force pilot, आपने देखे होंगे बालाकोट टाइम पे जब हमारा अभिनंदन साहब को जब कैप्चर किए थे, but particular kit is given to the everyone, right? This kit has containing the special type of food. It is called sometimes the survival food. It is called sometimes survival kit also. And fortunately, from DRD actually making this food for Indian soldiers for the survival food actually. This is known as a novel food category actually. These are the many ingredient which we cannot disclose also to you. It is comes for them actually. Even ready to eatable, ready to boil, ready to eat. This is all instant tea. It is comes. This is come in the generic food basically, but had to be used in the category of the novel food when particular application is there. Now, next slide. Now, let's really go from rule actually. I have listened the many rules. Now, next slide. Now, I just discussing what we have done actually. So only the published paper you can tell, because no other data I can share with you. I know that my limitations. One of the students, I know that your faculties also, Manas actually. He has done some good work in the basically beta cariophylline. Beta cariophylline is sometimes told as a nutraceutical sometimes, but beta cariophylline can be explored in a many way. Like it is a hollow energy they are giving basically. In obesity, basically we are using these cases, and obese type of your B57 generally generated. Basically, this can be available in the factor medicines. If you go to the three pictures. This is not. No. Now this is control actually. This is only the lung histopathology I'm showing. It is a very common. Just I'm explaining for only the one parameter here. This is just the control actually. If you go to the alveolar and the macrophagic positions, it's almost similar, very prominent. This is the alveolar sac actually. This is when the air is there, inside the air is there, and which will partially the changing your oxygen in between the layers actually, right? What is the function of the lungs? Lungs is basically the function of the exchange your O2 and CO2. That is the main thing. So if you go to the structurally alveoli, their membrane is very fine, and this only there is some diffusion constant is there. That's why if you go to the histo actually basically or alveolar, so alveolar forms will observe very well, because there is a very large amount of vacuoles of air will be observed. It is known as alveoli and alveoli, right? So basically this is a structure sub alveoli, and this small portion is alveoli. So this is all, it is a particular in control. Now this is a obese actually. Now obese, it is become the fatty, right? By using any molecule you can add in these cases. The obese, when you're using the obese mice, basically B57s or any SD also, SD rats also. Now this all small of these vacuoles and the collapses means it is comes together, right? So if you go to the volumes of the sac actually very high in these cases, but this could not be capable to exchange the air, right? It could not be exchange the air actually, because if you get a small of the alveoli, you will get the very good exchange, because surface area will increase. So in this cases lungs, if you decrease the surface area by any molecular pathogenesis, that's why obese have some respiration problems always there. Assam is they are getting the very big problem in oxygen exchange. Now it is the treatment of your BCP actually, that is beta cariophylline. Now see the structures of the alveolar is a very large thing in these cases, while you have this very small alveolar sections is there. But in cases alveolar all are large actually. It is a very collapses, right? So next slide. Now another papers also came this side giving the managed to acknowledge particularly again the beta cariophylline cross talk between AdtU and R1 AdtU R2 receptor basically. I think Manas was there. They can explain very well because he's done that time. Our fat is basically it is a white fat or brown fat. It is a brown fat, right? इसलिए बोलते हैं गलाना मुश्किल है ना. Now, if you changing the fat molecule to one molecule to the another molecule by exchange of the burning, by working, physiological working, the fat will be dissolved. Basically, fat will be changed. So there is a two receptor. One is called the AdtU1 and AdtU. So AdtU1 and R2 is very difficult to understand, separate each other. So we are getting the NFR and OH basically signal cascade, that's the nuclear factor, and OH is the signal path cascade. So compound 4080 basically give the obesity is a lung damaging actually. Now at the left side of the paw, if you observe, that is nothing change. It is a controller. This is you observe. It is a blue stripes, right? We are getting the more perfusion toward this one. If the obesity is not there and getting the fat molecules is taking this not perfused, that color will be not coming. So this means the first one highly non-perfused, right? You can go to the this one in the middle portion is the non-perfused because of high obesity, high fat. But in both cases, the caryophyllene actually preventing that the obesity and the particular diffusity of the AdtU1 AdtU2 receptor basically. So basically sometimes for the pseudo-allergic reactions, 48P is using for the pseudo-allergic reaction. There are these molecules as used in the chemical as a mimic chemicals. So yes, now do you think caryophyllene is a drug? Yes, caryophyllene, if you claim that it is anti-allergic, anti-obese, naturally it will become in a drugs. Otherwise, it is in the nutraceutical. Next slide. Now, the polarizations. I have told you, जितना सारे बीमारी होते हैं जो मोटापन का ही होता है, even I might be suffer, if you go to the obese actually, all the disease will become to you. But why? The excess fat, excess fatty molecules getting the inflammations. It is the highly inflamed actually. If you get the inflamed, they will change immediately in polarizations. So AdtU1 and AdtU2 is a two receptor, get the polarizations of the particular fat. So when the fat actually when it is migrated, it is called sometimes the Boyden chamber. Generally using the Boyden chambers, and we getting the adipocytes actually can collect the adipocytes, and then migration we observe from one chamber to another chamber. And this time all the molecules, all the fat, your molecule you can observe by your all the enzyme activity and the receptor. So in these cases we are many things we have observed like THP1, IF alpha, M2 marker. So M1 is a microphage 1 and microphage 2. And these conversions we can easily calculate it. I know the BCP 100 is your beta caryophyllene in 500 gram per ml. It is fantastic, challenging with that. This is a control one. There is a if you go to the M1 M2 factor, that is the PNA very high. In these cases, it is going to reduce. And if you go to the INF gamma actually, gamma is giving the protections. It is the somewhat protections we will observe. And in these cases, the control versus the BCP molecule, same thing. You will get the THP1 cells. You will observe the protections. Now this is actually another we are searching particular particular acute lung injury it is going on. I'm not going into the detail actually in this cases. We are taking the DA and AC2 receptor. In a COVID type symptom, COVID type your syndrome. AC2 basically your angiotensin converted 2 enzymes, basically 2 inhibitors. We are looking at the AC2. So we are taking the AC2 receptor and BCP and AC2 receptor with very good coincidence. And in these cases, in the molecular docking study, we are doing the new molecule we are taking in the beta caryophyllene derivative. So all study is done, almost the lung permeability, weight to dry function ratio, histopathology, all the respiratory dynamics actually. Respiratory dynamics also we have seen. And in this case, the CO2 O2 ratio, respiration rate, oxygen concentrations, we are all at the same. So this is about some research we are in part with this particular nutraceutical of the drug molecule. Next one. Now, just for some information to you, all the students is there. Fix the requirement to look at the deliverable. When you go to any research, think the what is the deliverable. Today is everything is changed scenario. And the research is does not mean your publication. Research does not mean your patent. So you will tell what is your product, end product. So I will tell you, fix the requirement of your what you deliver value. Second, never drink. Don't drink because every hard work pay you. If you do not do the hard work, nothing will be paid out. Today's world is not ready to give you anything without hard work. So not drink, irrational studies, raw material, unreadable lead compounds, and unrelated publications. Don't go through this. Fix your aim and go this area only, and you are established in your own area. Don't go other area. Next, don't go to the overextended. Third thing in the research. Even after my 15 years of the research career, I have seen that don't go to the overextended. What you are looking and what you target and what you are thinking, and go in. Know the step. What is your next step? What is the law is said, and what work according to the project you are allocated. Suppose I am telling you are going to the nutraceutical project. Don't ask the many things. You go to the law. What is the additives? As per law, add these additives. Make the some product and deliverable. That's how deliverable will become. Otherwise, nothing will become. Your law will not allow you. Next, pay attention, choice or approval process. How will go to the regulatory clearance? Today is the biggest challenge of any product. So you have fix your path and go to the same directions, and you will get it approval. Because your end product tell you the approval. You asked you the last product is getting your end form actually. It's in the table actually. So approval process is another important. 5A is always to remember in the research. That is avoid repetitive research and the brand challenge change. Don't go to the repetitive research and changing your brand. Avoid the different trade name and the some products. Avoid the survey which will not yield the deliverable. Rather fix the problem from the old survey and from other sources. So challenging is most challenging. What project will you do? What research will you do? So think for the what necessary for the society. So this is also another important for you. Focus your competitions with your competitor along with you, right? If you do not be a competitor, then you are not done your proper research work. You have not been a competitor. You do not know your who are doing with you, right? Next, listens for your consumer and user is says. What is requirement? Right? Like you are in faculty member, you are in student. Student is asked for the teacher what you are looking for me. Faculty member maybe ask from his head what are you looking from me. So what are you asking for me? And that should be your complete actually. Next one, think IPR protection that you save you later. So make any project forms, apply for the patent at least. Some publications also today, if you go to the good publications, and at least you can protect your IPR. Rather than somebody is going to the patent, it is I think the patent act is that better. I think you have to learn also. That is called the pre-grant opposition and post-grant oppositions. Even the patent is granted, but your publication before that, you can go to the oppositions, you can go to the grant oppositions, and you can retrieve the particular patent grant. So publish this also sometimes. Band arrogant uniqueness, like you are in DRDO, we are in the nuclear, biological, and chemical cloth, bioregistered Dabur brand product, Hull, the brand product, it is come sometimes as a brand arrogance. Now case study man, I will give the 3 case study to you. आप सोचो पेप्सी के साथ एक एडवर्टाइजमेंट है, आप पेप्सी पियोगे? नहीं. What it means of Pepsi? Pepsi giving me your rejuvenate means Pepsi पीने के बाद आप तीसरे मंजिल से भी छलांग लगा सकते. Pepsi is looking like that, right? You can jump from the 3rd floor like after drinking the Pepsi. So some people are sleeping after the Pepsi. Nobody will purchase it. The right productions, right time, and complete product to the servicing through the industry partner. This is another way to getting the project success actually. Next one, don't go to this one. Now what case study 3 actually, Kellogg's. I think the Kellogg's in India, they are telling that 2% of the share rate of the 18 million people, but Kellogg's is not getting the success in India. Why? What do you take? You prefer Kellogg's is recognized the breakfast, like the food, very culture, what people traditionally eat or India, breakfast different from UK or US. So our food is somewhat different from UK and US. So Kellogg's is failed in India. So know your user. If you know the innovation required user, then no free lunch. note your market potential. Nokia connecting peoples. Why the fail? Why the fail? Nokia had certainly understood that there is about the connecting people. However, had their brand promised that they have just been connected, they would have continued to leave more than relevant brand visions. So basically, that's why Nokia has failed actually. Yes, and 2009, the Nokia 48%. 2011, that is 29% because Android, they are not accept that point. So know your upgrade your technology and reunion is required. Nanocar is another fail. Why Nanocar is a fail? Tata Nano concept the upgraded family 4 and 2 year. Right? Every household had an aspiration to move to something better than necessary cheaper. Cost, space available in cheap, but actually is different in the Nano. It is not the 1 lakh car. Promotion never becomes a rural or middle income groups. Proper promotion in unique case and low cost product service to the industry partner is required. Ponds, if the Ponds make the toothpaste, the Ponds has came. Hindustan Liver came from the toothpaste. Nobody accepted because Ponds is made, but the beautification formulations. So that's why the Ponds is failing the toothpaste market because Hindustan Liver has a very big market in Hindustan. That's why in Ponds is fail. So know you are the brand actually. Like we are in the brand, so we are also doing some product in the making in the DR. A good regulator listens and listens. A good students also listen and listens, but act rationally as the asking questions for the approval. I think the IFL affects Morrison's and vegetable pill. When we see your pictures in the mirror, you can understand what I am and what you are, right? So research does not mean that you have done everything. But I think when see in the mirror, you can ask yourself what you have done today. So you can get a very good answer because self-reliance is the maximum you can realize it. So I'm not going much more. Thank you so much.
From the audience, anyone wants to ask any questions? If anyone wants to ask any questions, please come in front of the dais.
Yep, is that? Dosing of nutrition. See, dosing of nutrition. See, the nutritional dose is not so much. That is the equivalent to your daily food of intake. Right? Food of intake, you are taking almost in 2 parts also. Suppose you are taking 150 grams of rice or something, in maybe the 300 grams you are taking the food. So this way, nutraceutical should not be 300. It is at least 1/4. But nutraceutical giving the 4 division dose, and it can be your whole diet. Same as calorie value I have told you, that is 1,200 calorie could not be exceeded in your total. So basically, nutraceutical dosing is same as rule, but not as drug actually. So food, they can be complements with the food actually. Right? But if you take the along with the food, then you will take the special dietary or special food, like 50% of like dietary food. That kind of food can be supplemented.
Actually, many times we are this phytochemical, pharmaceutical. Is there any difference in between?
There is an interesting story. There is a phytopharmaceutical and phytochemicals. Even see that actually drug opposite 1940 and rule 1945. Say that for 29. Simply that is the use actually taking the middle role. And they have been simply from their books actually and I will photograph. I will cite the pharmacology. They have categorically mentioned what will be the animal product? Now, second part is something the phytopharmaceuticals. Phytopharmaceuticals obviously they are there in 2009. But nothing but for the new chemical entity. NCE. And phytochemical have the rules of 3 years. Any phytochemical which is replaced as an allopathic medicine, it is known as same as modern medicine and Number two, any phytochemicals usually used in emergency purposes, that is also known as the NCE, new chemical entity. And third thing is the modern pharmaceuticals that have to be ruled of the unknown. Because the all right.
Hello. 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 Any more query? Anyone is having any query or anything you want explanation? Well, thank you so much, Atwada sir, for your uh, incredible speech and incredible presentation. You have tasked very pertinent and very what do you call that very common topic and have explained to the fullest. I am sure that all the participants have thoroughly understood the smaller but important aspects of life. Thank you so much.
Now I would like to request Dr. Saikat Sen sir to please felicitate Dr. Damiki Lalu sir, Associate Professor, JIPS, Guwahati. Ladies and gentlemen, good afternoon. My name is Vishnuka Kaur, a student of the Pharmaceutical. Now, we are going to proceed with the 8th scientific session. Welcome to science. Various research teams. Dr. Reddy's professional journey began in 2000 as an apprentice trainee at the Madras Pharmaceuticals, Chennai. Since then, he has consistently pushed the boundaries of pharmaceutical research. His current role, which he has held since 2013, is as scientist too in the Chemical Research Department of Aurobindo Pharma Limited R&D Center, Hyderabad, where he has been actively involved in the process research of APIs, innovation, scale-up design, and chemical process optimization. Throughout his career, Dr. Reddy has not only excelled in the industrial sector, but has also contributed to academia. He served as an associate professor at Bharat Institute of Technology Pharmacy in Hyderabad and played a role in shaping the next generation of pharmaceutical professionals. Dr. Reddy's educational background is as impressive as his professional achievements. He earned a Doctor of Philosophy from Birla Institute of Technology and Science, Pilani, with a thesis focusing on the design and synthesis of gamma aminobutyric acid derivatives for the treatment of epilepsy. His commitment to research is evident through numerous publications, patent achievements, and conference presentations, showcasing his dedication to advancing the field of pharmaceuticals. Dr. Reddy's research publications span a wide range of topics, from anticonvulsant agents to antimicrobial compounds, highlighting his versatility and expertise. In recognition of his academic excellence, Dr. Reddy has received honors such as securing the highest dissertation marks in MPharm and MCOPS Manipal and being awarded a senior research fellowship from CSIR. Dr. A. Sunil Kumar Reddy is an accomplished scientist, educator, and innovator whose contributions have led to a lasting impact on pharmaceutical industry. His dedication to the pursuit of knowledge and development of new chemical entities exemplifies his commitment to scientific progress. It is with great pleasure that I invite the distinguished scientist and innovator, Dr. A. Sunil Kumar Reddy, sir, to join us on stage. Round of applause, please. Before we proceed, it is my privilege to invite our esteemed chairperson, Dr. Subhajyoti Sarkar, sir, Associate Professor, Faculty of Pharmaceutical Science, AdtU, to come up on the stage and felicitate our guest speaker. Now, I would like to request Sir to kindly proceed with the session.
Yeah, thank you for the felicitation and then nice introduction. Good afternoon to you all. So I hereby extend my reverence to our Dean, Faculty of Pharmaceutical Sciences. So I thank all the management, VC, Pro VC, revered professors, delegates, students, and I invite you all for my presentation. So at the outset, let me appreciate the teamwork done by the people, by the faculty or management for organizing such a nice conference. It's not so easy to do teamwork nowadays because people want credit individually. Okay? They don't want to own the people. Let me appreciate that. So before my presentation, I interacted with some of the students who presented their posters. So they were very enthusiastic. So I appreciate them. So I thank your management, Dr. Uttam, Dr. Saket Sen, for giving me the opportunity to share my experiences to you. So it is all at the end, students should get benefited. Then only your management will have an achievement that they will feel satisfied when the students have learned something or they've gained from something. Okay. So let me invite. So the topic. Working? That will be better. Yeah. Okay. Yeah. The title of my presentation is Process Chemist, the Challenges and Solutions for Drug Discovery and Development. So the presentation, the contents of my presentation are introduction. There will be introduction to the drug discovery. So what is the process? I will be giving an outline. So in that, principles of drug design. So what are the things you should know, or how you have to design a drug? What is the history? How the history drugs were designed? What is the current scenario? What are the things you have to know? I'll be giving an outline. Then I'll be talking about the types of drug design. So whether it is systematic or ligand based or okay. The next one I'll be talking about process chemistry in drug development. So what is the role of process chemistry? What is process chemistry? What is the role of him in the drug design and development? After that, assessment of pharmaceutical process. Suppose you are taking the process from lab scale into the commercial scale. So what are the factors? What are the things you have to know? How it will be impacting? So initially the medicinal chemistry route is different. Lab scale route will be different and commercial scale is different, and again pilot scale is different. Product will be same, but the process will be completely different. And then I'll be talking about impurities and nitrosamines. Whenever you are making drugs, there is an again big headache that there will be a lot of impurities and nitrosamines or PGI impurities. So how you have to synthesize them, or how you are limiting their formation, about that. And finally, PEPFAR and MPP. So PEPFAR is the President's Emergency Plan for fighting against AIDS, and MPP is Medicines Patent Pool. So this is called globalization. How they are helping to reduce the diseases. So coming to the introduction, maybe as a pharmacist, most of you know the drug process, right? So drug design, or it's a long process for a new drug. It takes around 12 to 15 years for a drug to reach into the market, and the failure chances are more. In the case of drug discovery, almost 80% failure rate will be there. So it starts with an idea, and project team will be there for assessing the costs and all. You start with the synthesizing compounds. Initially, you will synthesize lots of compounds. Then you will screen it, robotic screening or in vitro screening. Then in vivo screening. You will do the screening in vivo, whichever hit molecules are there. Hit molecules means if you synthesize 100, Around one or two compounds you may find active in vitro. They will be pharmacological active. So those compounds you will take for early clinical studies. So hit compounds means which is showing active in vitro. Okay, then you will get a lead compound. Once you do the screening in the in vivo, you will end up with a lead compound. Then you will go for the preclinical studies, formulation development, go with the clinical studies. So in the preclinical studies, you will be doing complete pharmacological toxicological activity in animals, so that you should reduce the number of drugs. Okay, you will start here with the thousands or lakhs. You will end up with 10. You will end up with 4, 5, and then finally you may get or you may not end up with a drug. So after clinical trials, you will go with the 3 clinical trials. I think 4 clinical trials: phase 1, phase 2, phase 4 phases of trials. After that, fourth phase will be the launching into the market. Then you will do the registration. Whichever country you are going to market, you have to register in that particular country. That is, then also you will do the post-marketing surveillance. So if you do look at the history of the drug discovery or drug design process, the olden days, before pre 1920s, there were only herbal discoveries were there. Some are serendipitous discoveries by accidental. They will be working on some other drug. You will end up with some other activity. So from 1920s and 30s, there was a discovery of vitamins and vaccines. In 1940s, antibiotic era started with the discovery of penicillin by Alexander Fleming in the year 1938. So it paved a way for the synthesis of lot of antibiotics. Okay, so R&D boost due to the World War II. After World War II, there were lot of loss of human lives. So there was a deficit of good potent drugs. Then it boosted the research into the drugs. In 1990s, robotics and automation have also come. So these modern techniques are reducing the time taken for the drug discovery. Yeah, this is the drug discovery process. So after informatics. So initially, drug discovery starts from the identifying the disease. First, you identify disease whichever you want to work. You want to work on tuberculosis. Yes, you identify. You have to first identify the target, right? Earlier, previous presentation also Chettopadhyay sir was telling target. First, you should know the target. Okay, whether usually it will be protein target. Isolate the protein involved. Find the drug effective against the drug. Then do further. So it will take around 2 to 5 years. Again, isolating the protein, it will take 2 to finding out the effective drug. Again, it will take 2 to 5 years. Do preclinical testing. It will take 3 years. Again, human clinical trials. Then finally, drug it will come into the market. Overall, it will take around 12 to 15 years. But here, in the using the informatics, you can reduce some time. You can identify the disease, identify the potential targets by doing in silico. Right? In vitro means outside the body, in vivo within the living organism, in silico using the system. Okay, you can reduce the number of drugs. Okay, do small molecular inhibitor in silico predictions. You can do instead of synthesizing and wasting time. You can reduce it. It will give a good idea. Then polymorphism also is playing a role, because whether it is crystalline or amorphous, particularly which form is having an impact on bioavailability. So you should have a knowledge on polymorphism. So again, electronic case report generation. So you can use the informatics and all these things. So informatics is reducing the time. So these are the principles of drug design. As we know that initially we should we should know our target, which one we have to work out. These are the broadly you can classify these targets into 4 types: lipids, proteins, nucleic acids, and carbohydrates. Example: lipids, drugs that disrupt the bilayer cell wall. lipid layer if they are disrupting some drugs. Example, amphotericin. And some drugs will act on proteins, act as enzyme inhibitors. These are AC inhibitors like captopril, enalapril, all pills. So some are receptor agonists, L-DOPA, antagonists. It is drug used to treat schizophrenia. Some will work on ion channels. Example, diazepam. So nucleic acids. Nucleic acids are also good targets for any drug design. Intercalating agents, dactinomycin. Alkylating agents, cisplatin. Okay, carbohydrates are also targets. They work by disrupting the structural function. Example is vancomycin. So you know what is serendipity? Serendipity is nothing but the accidental discovery. As we all know, penicillin was discovered serendipitously. Accidental discovery. Earlier days, nearly about 20-30% drugs were discovered serendipitously, accidentally. So example, astanilide and phenylbutazone were antipyretics. They are using antipyretics. Actually, one of the physician dispensed for anti-helmintic activity. He dispensed astanilide without seeing the label. Then they found out that his temperature was decreasing. So then they found out that these are antipyretics. Like that, each and every one. Penicillin is also antibacterial. Alexander Fleming, when he is working on Staphylococcus fungus, he found out this antibacterial activity of penicillin. Similarly, piperazine. So every class, almost most of the classes, some are the other drug serendipitous discoveries are there. So for if you are after knowing the target, suppose you have designed a target, some of the crucial features discovered by Chris Lipinski. He's a scientist in old scientist in Pfizer. He found out that, as we know, that many drugs will come and fail in the clinical trials. Phase 1, some will fail. Phase 2, some drugs will fail. Phase 3, some of the drugs will fail. Finally, hardly 1 or 2 drugs will reach. If you start with 1 lakh drugs, by the time it reaching to the human, hardly 1 or 2. So Lipinski, one scientist, what he did, he took all the data, why the drugs are getting failed, why they are not reaching into the market. He made some common rules. It is called as rule of 5. 4 rules, all are multiples of 5, so it is called as rule of 5. So they should have the fewer 5 hydrogen bond donors, OH, NH. These are all hydrogen bond donors. Okay, the drug should have less than 5 hydrogen bond donors, and it should have 5 hydrogen bond acceptors. Nitrogen, oxygen are hydrogen bond acceptor. The number should be less than 5. Okay, and the molecular weight is less than 500. The molecular weight should be less than, if it is more than 500, it may not pass the clinical trial. This is exceptional for vitamins and antibacterials. For them, the drug molecular weights will be more. Okay, the other one is partition coefficient of less than 5. Partition coefficient means it should pass through the thing. See, drug should be soluble. For getting a drug is very difficult. It should pass out at almost 20-30 features single molecule should be having. So drug should be novel. It should be patentable. It should be soluble. It should be bioavailable. It should be permeable. It should reach the target. It should bind the target. It should not have toxicity. It should not have anti-teratogenic effects. It should not be mutagenic, and so on. So 20-25 features single molecule we should achieve. We should make it. So it is hardly a very challenging task. Are some of the natural products. They screen upon some of the other diseases. Some animal models will be there. Anti-convulsants or anti-Alzheimer's disease, they keep on screening, which is not target, which is not randomly they will do. Okay, it is time consuming and it is very expensive, laborious, and low success rate. 1 in 1 lakh will be there. Now, after some time, as time passed on, rational drug design came. So it is not based on chance, it is based on reason. They will find out why that drug is active, which moiety is responsible. They keep on making the derivatives. So it may not involve computers here. But in the case of CAD, they will take the reason and they will do the computers they will use. They will find out the target in silico. They will try to dock it. They will superimpose this molecule onto the target and reduce the number of molecules to get synthesized. So this is the overall picture of the computer drug design. You should know that if you know the protein structure, know the structure of ligand. What is ligand? The one moiety which is binding to the receptor, that is called as ligand. Okay, the structure-based drug design, you know the ligand, know the protein structure, then you can do the structure-based drug design. If you don't know the ligand structure, you know the protein structure, still you can do the drug design. It is called as de novo design. Ligand-based drug design, you know, don't know the protein structure, you know the structure of ligand. So you can find out what are the common features all ligands are having. From that you can design the drugs. When no known ligand and no unknown protein structure, you cannot synthesize anything. CAD is of computer-aided drug design is of no use. So this is all the overall picture of computer-aided drug design. The structure-based drug design, you will know the structure of ligand as well as the ligand means drug which is binding to the receptor. Okay, you will have the structure-based drug design. You will find out the binding site, which site is binding to the target. Then you will do the docking and scoring, which you will make several structures. Some 100 structures you will make, and you will dock onto the target. You find out the binding site, what are the common features. From that, you can do the scoring. You can give score, normal score, out of 100, how much score you can give. Do the virtual screening, do compound selection, then you do the lead optimization. Lead is nothing but the one basic structure from which you can modify the analogs. Then you will get the new drug. In the case of ligand-based drug design, ligand-based means you will be knowing the structure of ligand, you don't know the structure of target. So from several ligands, you can find out the common feature and then do the QSR is also part of ligand-based drug design. Do the pharmacophore modeling. Pharmacophore in a ligand, whichever is binding to the site, that is called as pharmacophore. In the complete molecule, all molecule will not bring the pharmacological action. Complete molecule will bind to the receptor. Some part of the molecule will bring the pharmacological change, will bind to the site, and it will be acting as agonist and antagonist. That is called as pharmacophore. The molecule which is not having any impact on this is called as oxophore. Okay, ligand, pharmacophore, oxophore. Okay, so here also we'll reduce the compound selection, do the lead optimization, you will end up with the new drug. So I will tell what is lead compound again. So these are the normal databases, whichever databases means this. Every time you did not have the structure, they will be having thousands and millions of the structures, common structures. These are the common databases, link database, Campbell, JChem for Excel. Every country is also having softwares and databases already pre-designed, pre-drawn structures. So you can directly take the structure and do the docking studies. So some people will tell some people what is the use of computer-aided design? Are they successful? Have any drug have reached to the market? So these are the examples. Captopril, as discovered by BMS, Bristol Myers Squibb. It is an AC inhibitor. This drug is designed by using computer-aided drug design. Okay, so dorzolamide is also a carbonic anhydrase inhibitor. It has also reached the market where computer-aided drug design has reduced the time. They have done this through the computer-aided drug design. Saquinamide is also a HIV protease inhibitor. Janamivir is also a neuroamidinase HIV inhibitor. Aliskiren, renin inhibitor, antihypertensive agent. Osipravir is a hepatitis C virus inhibitor. So in CP, it is in maybe development, heat shock protein inhibitor. Okay, so these are the drugs which reached into the market using the computer-aided drug design. Okay, so this is all the part of medicinal chemistry. Medicinal chemistry is different again. Process chemistry is different. Medicinal chemistry initially he will design, and for he will be synthesizing the compounds in a smaller scale. Process chemistry will do in a larger scale and commercial scale, because again the chemistry part is completely will be varying. So process chemistry is nothing but the branch of pharmaceutical chemistry. Okay, that is refers to the design and development of synthetic routes for the ultimate goal of manufacturing at the commercial scale. Medicinal chemistry, a person will do at the smaller scale, milligram scale or microgram, or the maximum gram scale, because he don't want more compounds. He will synthesize, keep on synthesizing, do in vitro study, get the hit molecule. Okay, he will synthesize hundreds, thousands, get one or two hit molecule which is in vitro, active in vitro. Then that molecule he will take to in vivo. If any molecule is active in vivo, then that is called as lead molecule. Lead molecule, from that he can make the structural modifications. I will show the structural modifications what are you can make. So The goal of ultimate process chemist is safety is more important, because lab scale you are working on smaller scale, it will not have any impact. If any reaction explodes or something, it will not have any impact. But for process chemist, safety is the most important. Durability or robustness. Robustness, process should be robust. When I perform or anyone can perform, it should be repeatable. Okay, robustness important. It should be eco-friendly. Environmental have. Cost is also an important factor. Okay, you should work on. You should produce the compound with in a smaller scale using a cheaper reagents, cheaper solvent, and quality is ultimate final destination. Quality, without quality, nothing is there. Okay, so goals are safe, cost effective, reproducible. It should be robust and it should be suitable for scale up. So process chemist and medicinal chemist will look at the diversity of molecules. You want different molecules. You want to change different structures, so that you should find out the activity, which structure is having what pharmacological action. Here he won't look at the scale up issues and all. Whereas process chemist, he will work on only one molecule. Okay, ultimately he will completely change the synthetic route. Here medicinal chemist, whatever route he is making, here it will be completely different. Okay, he will target only on one molecule. He will improve the process. Okay, so process chemistry initially started in 1950s. It emerged as a profession. In 1950, it started Gordon Research Conference. Gordon is a professor in the United States. He's a process chemist. So it started in 1954. So this is the evolution of process chemistry. Initially, 1688, MEP started process company. In 18th century, there were some other companies, Roche, Bayer, ICI. In 1880s, launched the first pharmaceuticals. In 1914, Eli Lilly implements the first written SOPs or guidelines. 1932, Bayer launched the first commercial antibiotic. 1950 to 53, MEP synthesis of cortisone, which is a milestone in the process chemistry. So if you see the timeline where the process chemistry, he will not involve in discovery. Of course, he may support for the scale-up issues. Okay, so preclinical development also process chemistry will be involved. Exploratory development. After developing, there will be a lot of toxicological studies where you will be needing the good amount of quantity of the compound. There also process chemistry will play a role. In development, life cycle management, and launch also process chemistry will have a role. So if you see the product, see initially linear synthesis means directly do A plus B plus C will give D, will give F, so it is linear. So in step 2 linear synthesis, they will take the raw material, reduce the intermediates. Here 6 intermediates, 4 intermediates, here also will get the product. In convergent synthesis, they will take the different parts, part A to B, and then they will combine. So here the cost, in the linear synthesis, it is route 1. The cost is more, yield also will be less. Cost is more, yield is less. In the route 2, yield cost is less and the yield also will be more. In the case of route 3, the cost is less and the percentage of yield also will be more. So divergent synthesis will have is beneficial than the linear synthesis. So this is the involvement of process design. You should have knowledge in synthetic organic chemistry, physical chemistry, commercial chemical engineering, analytical chemistry. You should have knowledge in safety, health, and environment. And crystalline science, you should have an experimental knowledge in statistical design. So this is the overall expertise a process chemist should be needed. Okay. So for the process chemist, they should focus on all these things, like select criteria. The process should have all these criteria. It should be safer. Okay, safety is more important. As I said, whenever you are transferring the lab scale hardly you are doing 10 grams. Pilot scale, you will be doing the same molecule in kilogram scale. Okay, but the completely route may be different. They will use different reagents, different starting materials, product, but the product will be same. Okay, so usually the generic drug and innovator drug. Innovator's route is medicinal chemistry route, whereas generic drug, you will take up a different route, but with the same quality, same parameters. So whatever innovator is achieving, generic version also will have, but the price will be less because after patent, he will be synthesizing these drugs. Okay, so safety is more important. Environmental, all the routes should not have impact on the environment. And legal means it should not have infringement to the patent. Suppose someone has patented that route, you cannot use the same route for marketing your API. You have to use a different route. So this becomes illegal. Then you should not use whatever the chemicals banned. You cannot use in the process. Medicinal chemist can use. For him, legal things will not have because he is using on smaller scale. And similarly, economic. So economic is more important, right? You are using chemicals in kilos, your price should be lesser. And control of quality is important. If you are working on enantiomers, diastereomers, you should get a single compound. So it should meet the expectations of GMP. Through you should manufacture the drug in a limited amount of time. Okay, so time scale of manufacture. So long synthetic route, rare natural products, it will take more time. Throughput means it should reduce the time, the amount of time you are making for a particular API, active pharmaceutical ingredient. So this is the outline about the process. And along with the process, there are several other issues. So it's not that medicinal chemistry are synthesized, so only you are reducing the price. Only cost is not the criteria. There, as I showed, we should follow the select criteria. Okay, you have to have a knowledge of impurities, nitrosamines, PGIs. Okay, so many things a process chemist has to be. He should be thorough about, and he should have knowledge on regulatory bodies. Okay, and he should fulfill the needs to launch a drug into a particular country. Suppose you want to market a drug in US, you should get it approved by the US FDA. In India, it is by DCGA, the Control General of India. Every country is having a regulatory agency, right? You should get it approved, registered from them. They will have a committee. They will, after once you file a drug, you have to apply a DMF, drug master file. You will give a complete information. What is the route? What are the things you have given? Then their committee will go through that. Then they will approve it. Then only you can market that drug. So impurities. So every drug will have impurities. Suppose you are making paracetamol, it cannot be 100%, right? At the most, it will be 98% or 99%. Other things are impurities. Impurity is nothing but the chemical structure other than the API, active pharmaceutical ingredient. So impurity is defined by the drug substance by ICH, International Conference on Harmonization. Till 2015, it is International Conference on Harmonization. After 2015, it is International Council of Harmonization for pharmaceutical requirements for technical requirements for human use. Okay, it is not as per them. It is a drug substance that is not the chemical entity. You call it as impurity. So impurity profile is nothing but the impurities. Identified unidentified impurities present in a drug substance. So drug substance is nothing but the bulk one. The product is a completed form, finished drug. Okay, you can broadly classify the impurities: organic impurities, inorganic impurities, residual solvents. So these impurities can be starting materials. They will get carried. Suppose your API is 5 steps. You can start from the initial one. Starting material will get carried till the end of the API. So these are starting materials. Byproducts. Some of the byproducts released in your while making API. So those are also can be a impurity. Residual solvents. You are using solvent for making API. So for that also there will be limit. Astronitrile, it should not exceed 780 ppm. DMSO, it should not exceed 4000 ppm. So it should, an API should adhere all these things. Okay, like this, there is more than 100 criteria, 100 tests it should pass once the API has to come into the market. Okay, so residual solvents, intermediates, some of the intermediates will get carried through till the API. Degradation products, some of the things once API is manufactured, it will get degraded. If you are keeping in the air, it will get oxidized or it will get hydrolyzed. Okay, reagents, ligands, and catalysts. What are the reagents you are using? What are the ligands, the catalysts? All these things it will be carrying out. Heavy metals, inorganic salts, other materials are also carrying out. It will get carried till the API. One second. Next one slide. One minute, one slide I missed. Some slides. Back my arrow. 1 minute. Repeat slide. Yeah, 13. No, back 9. 12, 13. Okay. One. Huh? नहीं आ रहे.
Yes, sir.
नहीं. Okay. नहीं आ रहा है. Structures. Put in वाला आ रहा है. 12, 13. Okay. Okay, that's what I. Structures नहीं आ रहा है. I missed that. 12, 13. Okay, okay. Next you go. Okay, then I will see. Footing, I will go to the. Okay, I think structures are not coming. Okay, इधर आ. ठीक है चलो. Sorry. One slide I missed. That's what I was. Okay, leave. So these are the broad classification of impurities. Nitrosamines are also impurities. I will show the next slide. So these are the residual solvents. Whatever solvents you are using, for that also ICH has classified that you should not exceed, APA should not exceed this. You cannot use these solvents, class 1 solvents, benzene, CCl4, 1,2-dichloroethane. They are highly carcinogenic, cancer causing. So you cannot use those solvents while making API. And well, for class 2 solvents are acetonitrile, chlorobenzene. So it should pass these limits. It should not have more than 410 ppm. Acetonitrile should not exceed 410 ppm. Chlorobenzene should not exceed more than 360 ppm. Class 3 solvents, potential daily exposure should be less than 50 mg per day. In class 4 solvents, there is no toxicity data. We can use them. And genotoxicity, a compound is said to be genotoxic when it modifies our genetic this thing, when it brings out mutations, when they are carcinogenic. Those impurities are called as genotoxic impurities. So when a compound, how to find out whether a drug is genotoxic or not? Using a softwares you can do, like docking studies we are doing. For the impurities also some softwares are there, like Derek, Lara, and Sarah, Derek, Sarah, lead scope. These are the softwares. By drawing the structure, it will give an idea that whether compound is genotoxic or not. For that, again, in vivo test is there, Ames test. So you can find out whether they are using this is a microbe, whether the particular compound is having genotoxicity or not. So these are the functional groups which may be genotoxic. Hydroxyaryls, N-acetylated aminoarylated alcohols, aldehydes, N-methyl. These are the compounds which may be genotoxic, which will alter, which will create mutations in our body, and it may lead to cancer. Okay, these are the functional compounds, and these impurities we have to make a strict control. Like it should be lesser than a ppm, lesser than ppm levels, not micrograms also. Okay, so if you see some of the API also will get converted to the impurities, like degradation impurities. Suppose benzoperoxythiophene, upon air oxidation, here it will get oxidized and form a keto compound. In the case of dexamethasone, so these alcohol CH2OH, it will get converted to keto compound. Similarly, emtricitabine, sulfur will get oxidized to sulfoxide. Okay, so all these impurities also you have to synthesize, and a process chemist should synthesize and find out the RT and RRT of HPLC. What is the percentage of this? Okay, esomeprazole, it is sulfone. So sulfoxide, this is sulfoxide, single sulfur oxygen. 2 oxygens, sulfone, esomeprazole sulfone impurity. Physoteridine, here also this alcohol is getting oxidized to acid. So this is also an impurity of physoteridine. Phenolcetran is N-oxide impurity. Nitrogen is getting oxidized, aerial oxidation. And perchloroperazine, sulfur is getting oxidized to sulfoxide. Quinacrine, nitrogen is getting oxidized to N-oxide. Rabiprazole, it is getting both impurities, sulfoxide, N-oxide. Tolteridone also, N-oxide is there. So all these impurities you should characterize, and these impurities should be lesser than the specified levels in the final API. So this is the lomestine. It is an Alkylating agent, maybe you all knowing all these things. It is an anti-cancer agent. So C-lomustine upon degradation, it results in simple cyclohexylamine. Lomustine upon hydrolysis, you will get an acid here, carbamic acid, which upon loss, upon heating it, by natural heating, of temperature exceeds 40 degrees, it will lose carbon dioxide, it form cyclohexylamine. So drug loses its potency. Lomustine loses its potency upon degradation. So these are some other API degradation impurities, chloranphenicol, where this chlorine is getting substituted by hydroxy compound in the presence of moisture, air moisture. So this is a fluorphenicol impurity. Arnidazole, also it is chlorine is getting substituted by hydroxy upon aerial oxidation and moisture. And amlodipine, here this chlorine is getting dechloronized. And atorvastatin, here this chlorine is getting substituted. Okay, this chlorine impurity. So each and every drug will have several impurities. If you read the pharmacopoeia, IP or USP, every drug at the end they will give the impurities, how to characterize it, what is the method, HPLC method, and everything we should know. We can check the purity with the help of HPLC, and the limits of these impurities should be lesser than the specified levels in the pharmacopoeia. So these are some other impurities. Diclofenac, okay. And upon diclofenac, it is getting loss of water molecule, getting cyclized. This is the impurity, cyclized impurity of diclofenac. This OH and here hydrogen will lead to the loss of molecule, get cyclized to get this moiety. Flupretrin. Here also there is a cyclization. This amide will get here this is hydrolysis upon acid and presence of acid and water, ester is getting hydrolyzed to acid, and this upon water removal, it is forming this impurity. This is getting cyclized here. Okay, keto and two amine groups. In the case of prunapril, here also this impurity is forming diketopiperazine. Here it is getting cyclized diketopiperazine. So this is a quinapril impurity. Not only in quinapril, all prils will having this common impurity. Diketopiperazine impurity, they will get cyclized very easily. This is the most frequent impurity in all the prils, quinapril, enalapril, tantalopril. So all these are AC inhibitors. So these are some other impurities. And arnidazole, it is forming a epoxide. It is getting cyclized. This chlorine is taking this hydrogen. Oxygen is taking this hydrogen. Loss of chlorine molecule, they get this oxygenated product. This is a major impurity in arnidazole. Similarly, jofinopril, this is getting hydrolyzed, or this acid group is converted to OH group. No, this slide. Actually, chemical schemes I have drawn. Yes, schemes are not missing. Schemes are like supported. It is not supported. It is word version only. Yeah, I have drawn some schemes, synthetic schemes. It is not basically. Okay. The previous slide is not supporting, not coming. Okay. Next one. Okay, whatever structures I pasted, some are coming. Whatever schemes I have drawn using software, that is not supporting. Yeah, next slide. Okay, leave it. So this is the normal process flow of a pharmaceutical company. So initially they will start with a literature search. Process chemistry again different. Medicinal chemistry, they might have launched the drug. Okay, drug will be already into the market once the drug is off patent, or even if it is in the patent, they can file. They can do the same drug with a different route, whatever the patent has been not covered. Okay, so they will do the product selection. Project people will select which project we have to work. Then they will do the literature search and IP evaluation, because someone else have patented. You cannot do the same route and you can market. You have to do in a different route. You should not infringe the patent. Okay, you have to select the synthetic scheme, which scheme is suitable, like following the select criteria, safety and all those things. And you have to follow whichever is getting lesser cost of. You should choose the lesser chemicals, smaller synthetic steps. You have to use all these things. Okay, and then you select the synthetic routes. Do the feasibility studies. Feasibility studies are nothing but find out the best study. For each every compound, there may be hundreds of routes, but it should go to the market commercial scale. You have to do the scale up. For that, some routes are not suitable. So for making a single compound, 1,000 routes will be there, but for commercialization, making one compound, it will be one route. It will be very, very difficult. Hardly, it is practically impossible. Making, because for commercialization, I told you there were several factors you have to suffice, you have to fulfill it. So it will be very challenging for a process chemist to make a to commercialize the product. So do the feasibility studies, do the analytical method also. Analytical method is important. You should know the RT of a drug. You have to synthesize all the impurities. We have seen the series of impurities. You have to synthesize them and give it so that this is APA, this is the impurity, this is the level. You should know all the impurity structures, APA roots and all. Okay, process optimization. Do the optimization, synthesis of impurities. Optimize it. Optimization means you have to do several reactions. Same functional group interconversion, but 100 routes, 100 reactions you have to keep. You have to use this much equivalence exactly. For 1 mole, you have to use 1.5 mole equivalence. Optimize the temperature. Optimize the good suitable condition. That is called as optimization, right? This much time. I have to stay for this much time only. I have to use in this scale. I have to remove the impurities in this method. Okay, do all the process optimization. You synthesize all the impurities, possible impurities. Okay, during the process also several alternate impurities will be forming. So you have to synthesize and give it for the analytical department for the method. They need the structure, everything. Okay, so they will develop the analytical method. Do the concurrent pilot batches. Once you are doing the lab scale, you are optimizing the process in the lab. Go to the pilot plant. Pilot plant is nothing but intermediate between the commercial scale, large scale. Pilot plant, it is like half a kg or 1 kg. Okay, then kilo lab scale, okay, up to kg. Pilot plant from kg to 100 kg. Then commercialization, it will be doing, you will be doing in tons, right? You are supplying APA throughout the countries. Then you need in tons. Every batch you will get 1 ton. For that, completely route is different. Again, you have to do validation. Validation at the lab scale, you have to do validation. You perform the 3 trial batches, 3 batches without a change in the, not more than 5% variation in your synthetic yields, process, quality, everything. Similarly, once you do some pilot batches you have to give for the formulation people, right? They need some bigger quantities so that they can also do the trials. Formulation people, then you have to go to the plant. You have to do the plant validation. Plant, it will be continuous. It will work on shifts. General shift A, B, C shifts, continuous 24 hours you will be working. You have to do the validation in plant also. Then you will go to the filing. Filing means you will show all these 3 validation batches. You will be completely giving this information to the filing agency. If you want to market the drug in India, give it for DCGA or some other FDA for US. And sometimes you have to do as per the ICH, International Council for Harmonization, because ICH, around 60 countries are members in that. So it is a common platform. So to avoid testing, unwanted testing in all the countries, there is a common testing will be there. It should ICH compound, it should be 98% pure. Any unknown impurity should not be more than 0.1%. Known impurities can be up to 0.2%. You should not have diastereomers. If there are chiral impurities are there, it should not exceed 0.5%, not more than 0.02%. So all these limitations, if you are marketing that, you have to make an active pharmaceutical ingredient by meeting all these specifications. Every intermediate will have a specification. You have to design your own spec. Okay, since the compound, you should say that this impurity should not be more than this level. So same compound, it may be multi-step synthesis. Okay, do the concurrent pilot batches, process validations, and even validation should be there, done at the synthetic lab, and the analytical people validation also should be, they should do for each and every APA in the lab scale as well as in the plant scale. And then the stability studies, they are also, you have to do for each and every intermediate as well as to the final APA, you have to do the stability studies. Then only you will go to the drug master filing, DMF filing after that. So for API, it is DMF, drug master filing. For formulation, ANDA, for ANDA, abbreviated new drug application. Why it is abbreviated? For a new drug, they have to do clinical trials. Okay, clinical, they have to do the clinical trials in small mice, animals and all, that is called as preclinical trial. Initially, in vitro screening. You will do the screening for in vitro. Whenever they are active, go to the in vivo screening. Those are called as a lead compounds. If any compound is active, after doing in vivo screening, do the extensive toxicity studies. Then you have to go to the clinical trials, phase 1, phase 2, phase 3. I think all of you know the clinical trials. Phase 1, some less than 100 people will be tested, 100 patients. 100 healthy volunteers. Phase 1, it is healthy volunteers. They will check the safety of a particular API. In phase 2, it will go to the up to 1,000 people, 1,000 patients they will be taking. And here they will check the safety and efficacy also. Phase 3, it will go more than 1,000 patients they will check. Again, they will confirm this drug is safety or efficacy. After passing phase 3, then they will go to the launch. They are going to register that particular drug in a particular country. After that, also they will be doing post-marketing surveillance. So, in the case of ANDA, so someone has filed all these drugs, they will patent it so that up to 20 years they can market. From the date of the patent, up to 20 years, no one can market this drug. Once the patent is expired, so if you are changing the formulation, you no need to do the clinical trials. So it is called as ANDA, abbreviated new drug application. So at the end, it is step for president's emergency plan for fighting against AIDS and medicine. Okay, medicines patent plan. So in the US President George Bush has started in 2003. So why I am telling this is like process chemist or medical chemist should have a complete thorough understanding in all these things. Okay, you should be having knowledge on process chemistry or globalization, analytical technology, even QBD is also there, quality by design. So don't test at the end. While designing itself, you should have a quality by design. You should have a knowledge on process analytical technology. The process chemist should have a knowledge on engineering and so on. Okay, so this is a PEPFAR plan was started by George Bush and his wife Laura in 2003. This is for low-income countries like Ethiopia, Kenya, and Haiti. This US has started funding to encourage research on AIDS and sometimes it's malaria and tuberculosis. For these diseases, these countries are poor countries. So the US government started funding on this so to encourage the researchers to find out the new drugs. So these people have helped millions of lives. They saved a lot of lives. And one more is MPP, Medicines Patent Plan. Here, Medicines Patent Pool, Medicines Patent Pool. Suppose patent holders will have, if you want to market, you have to pay to the patent holder. So what these people do, Medicines Patent Pool, it is also started in 2010. It is a United Nations funding agency. What these people did, they will talk to the patent holders. They will they will give funds to the patent. They will get the royalty rights, and they can synthesize these drugs. They can do research. So this is also to treat HIV, TB, and malaria. So patents are intended to reward. Simply patenting and keeping it is of no use. So if you sell for someone, all the other people, researchers who are working on HIV, TB, they will form a pool and they will pay to this money to the patent holders, and then they will also get the reward, and we can get innovate new drugs for the people. So this is also good plan, good strategy. So this is a small quote about the process people. For a process chemist, it should be process chemist means one armed person should pour and remove it. Like in lab, you can do scratch it and everything you can do. For a process chemist, you should pour it. You should nothing will happen directly. You should get the compound. Process should be so much easier. That is the quote given by John Carford. So this is one more nice quotation about Albert Sajin. He is a Hungarian biochemist. He is a Nobel Prize winner. He is a late person. So he says that discovery consists of what everybody has seen, but thinking what no one has thought. You can see the same API. You can read all the things what he has done, but someone have missed some thought. You think it and start working. So with these few slides, some of the synthetic slides I missed because they were not. At least I could have shown. Okay, it is not supporting here. Okay, I drawn the synthetic roots of whatever in Aurobindo what we have worked on cephinamide mesylate, is an anti-Parkinson disease, and acetylcarbamazepine acetate. Two schemes I have drawn. How much time it took for making these two, and we have taken from lab scale to the large scale these slides, and some history masterpieces I have drawn, like masterpieces how the medicinal chemistry route is different from my process chemist. Some slides are not visible. These are nothing but the schemes only. Maybe if I have shown also, it may be boring for you people. It is completely functional group interconversions. Okay, so you can see there are a lot of masterpieces in process chemistry. So with these few words, I thank the AdtU and the students and delegates for giving me opportunity and for patient learning. Thank you very much. Yeah.
Now we are open for questions. I request you all to participate actively. Anyone?
Any questions? Any questions? So you understood what is medicinal chemistry, computer-aided drug design, process chemistry.
I think it's crystal clear, sir. Thank you so much, Dr. Reddy, sir, for sharing your expertise and invaluable knowledge with us today. And here we conclude this incredibly educating scientific session. Thank you all for your active participation in this session. Now, I have an important announcement to make. It's time for us to take a well-deserved lunch break for 30 minutes. We will reconcave after the break to continue with today's event. So please enjoy your lunch and be